Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Examiner Dr. Endang Astuti MSI. Thank you for giving me the opportunity to present my undergraduate thesis arranged under the supervision of Professor Dr. Winarto Haryadi MSI and Dr. Rerum Naturalium Gian Primana MSc with the title Chemical Profiling of Secondary Metabolites Produced by Endophytic Fungi Isolated from Aglaia foveolata using Liquid Chromatography High Resolution Mass Spectrometry. My name is Setya Kusumawati and here is my presentation. Almost all the antibiotics used as medicine today were discovered and developed during the period from 1940 to 1960. However, bacterial resistance to antibiotic treatment today has become increasingly common. AMR was associated with 9.95 million deaths worldwide in 2019, with 1.27 million deaths directly caused by drug-resistant bacterial infections. This makes AMR a leading cause of death globally, comparable to major diseases like HIV/AIDS and malaria. To overcome bacterial resistance to antibiotics, new antibiotics are needed that have a different structural framework and a different mode of action from those currently in use. The structure of new compound is more likely to be isolated from sources of microbes that have not been explored much, one of which is endophytic microbes. One promising source is endophytic fungi. These fungi can produce beneficial chemical compounds, including one that fight bacteria and viruses. This is especially important today as more bacteria are becoming resistant to antibiotics. Aglaia foveolata, an Indonesian endemic plant from the Meliaceae family, harbor endophytes capable of producing unique and pharmacologically potent secondary metabolites. The purpose of this research is to isolate, characterize, and perform screening of endophytes, secondary metabolites from endophytic fungi, Aglaia foveolata on various media, and to determine the potential antibacterial activity of compounds isolated from endophytic fungi, Aglaia foveolata, against MDR bacteria. Hypothesis formulation. Several compounds isolated from Aglaia foveolata stem bark, particularly 3 terpenoids and steroids, have demonstrated significant antibacterial activity against both standard bacterial strains and multi-drug resistant pathogens. Fermentation-based technique has been explored to increase yield and alter the compound profile, yielded pharmacologically important compounds, including silversterol and its analogs, alongside a variety of 3 terpenoids and steroids known for their antimicrobial and anti-cancer properties. If the endophytic fungus Aglaia foveolata is fermented and its mycelium extracted, it will produce secondary metabolite compounds of the terpenoid and steroid groups which have antibacterial activity. Hypothesis formulation 2. The minimal inhibitory concentration is the lowest concentration of an antimicrobial agent that inhibits visible growth of microorganisms. Certain compounds isolated from Aglaia foveolata showed activity against Pseudomonas aeruginosa, Escherichia coli, Staphylococcus aureus, Bacillus subtilis, and Klebsiella pneumoniae, with minimum inhibitory concentration ranging from 29.4 to 125 micrograms per milliliter, indicating moderate to strong antibacterial effect. If the secondary metabolite compounds isolated from the fungus have antibacterial activity, then when tested on MDR bacteria, it will produce moderate activity to significant MIC, which is less than 10 micrograms per milliliter. The research was conducted in several major parts to prove hypothesis 1, the preparation of the sample including fermentation and extraction, and analysis including thin layer chromatography, flash chromatography, and high-performance liquid chromatography. And to prove hypothesis 2, it is comparison of isolated secondary metabolite with database and antimicrobial activity tests were done. Full research design are shown in this figure, and all of the experiment and characterization were carried out in the Research Center of Chemistry, National Research and Innovation Agency, Indonesia. Fermentation and extraction. The fungus was fermented in an Erlenmeyer flask with YMB medium. YMB medium was made with a composition of distilled water, glucose, yeast extract, and malt extract. Erlenmeyer flasks containing media and fungi were incubated in a rocking incubator at 120 rpm, 29 degrees Celsius for 20 days. The metabolite compounds were extracted from the mycelium formed after fermentation. Extraction was carried out by macerating the mycelium with methanol for 2 hours, followed by filtration to separate the extract and the mycelium. The extract obtained was dried using a rotary vacuum evaporator and was put in an oven at the maximum temperature of 50 degrees Celsius to yield a crude extract. Flash chromatography. The separation of crude extract was carried out using a preparative flash HPLC instrument with a Bushi revolveris, silica column, and solvent, which is chloroform, acetone, ethyl acetate, and methanol. Prior to separation, the extract were profiled by analysis using HPLC to determine the UV absorption profile of compounds, which would later be used for compound detection in preparative HPLC. The extract was injected by impregnating it in silica gel. LC-HRMS. Pure isolate were analyzed using LC-HRMS. The results from the search database were viewed for identified compounds from the analyzed genera with molecular or mass formula that match with the LC-HRMS data. Each match was investigated by comparing literature and experimental data. Professional compound assignments were made when matches were identified. Antimicrobial activity test. Pure isolate was added to the bacteria in each tube, which contained Escherichia coli, Bacillus subtilis, Staphylococcus aureus, Pseudomonas aeruginosa, and Klebsiella pneumonia. It is then put in a 96 well plate and the determination of MIC value can be determined. There are 12 fractions that can be analyzed, and fraction 10 is the most active due to the retention time peaks appearing on the chromatogram of the isolate, which indicate the presence of another compounds beyond the peak present in the blank chromatogram. The high-resolution mass spectrometry chromatogram of fraction 10 at a retention time of 8.10 minutes shows a distinct fragmentation pattern with peaks at mass-to-charge ratio 522, 496, 309, and 236. The peak at mass-to-charge ratio of 496 is identified as the base peak with the highest intensity, and it is presumed to correspond to a derivative of polyporinic acid and a mass-to-charge ratio of 486 or polyporinic acid C and mass-to-charge ratio of 483, which is possible with the addition of CH or CH2 groups. A fragmentation pattern for polyporinic acid C with mass-to-charge ratio of 309 was also observed, supporting the assumption that fraction 10 contains a derivative of polyporinic acid. A more definitive structural analysis would require further elucidation using proton and MR spectroscopy. This is the lanostate triterpenoid structure of polyporinic acid A and polyporinic acid C, which is obtained from fraction 10. Antimicrobial activity test. Presence of hydroxyl groups in lanostate triterpenoid derivatives is believed to significantly contribute to their bacterial activity and may enhance the compound's ability to interact with bacterial cell membranes or target essential enzymes, leading to cell disruption and death. This finding highlights the potential of lanostate derivative as promising scaffolds for the development of broad-spectrum antibacterial agents. It can be concluded that the purification results of the extraction obtained 12 fractions, with fraction 10 as the most active fraction. And from the result of researching the proprietary database in the Dictionary of Natural Products on USB in 2021, it is found that the compounds contained in fraction 10 are lanostate triterpenoid compound, extract from endophytic fungi isolated from Aglaia foveolata plants, have antibacterial activity. Suggestion: fermentation treatment using several different media are needed to influence the growth of endophytic fungi, and the toxicity testing of the compound is necessary for further research into the use of the obtained compound as antibiotic precursors. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.